All right, so first thing, I wanted to mention a few things about the lab module, and I wanted to remind everyone that the lab modules are at the bottom of the course. So you have to scroll all the way down to the bottom to see the lab modules. And lab module 1 includes a lab safety quiz, a microscope lab report, which is now live. I took out the part about the growing the mold because students had some trouble. Some of my students had already tried to do it and they're having trouble getting the mold to grow very quickly. So I took out the mold portion. And I'm going to be adding a very short, really brief microscope quiz. It's just like, what are the parts of the microscope? What are their functions? They're all listed in that booklet that came with your microscope. And then, yeah, so it's mostly just how to use your microscope and some characteristics of microscope. So that quiz will open today, later today, and it'll be open probably till Friday so that everyone can get it finished. I would just, again, remind you that if you can get that lab safety quiz and the microscope lab done before spring break, great. If you want to do it during spring break, great. The due date is, I'm putting a due date on it of April 30th because I'm just going to kind of leave it open for people who don't get it done. I'm trying to push you to get it done by this Friday so that you're off next week. And also, as a reminder, when you start growing the bacteria, you need to be able to babysit them every day. And I purposefully didn't want to plan you to have you start doing that until after spring break, unless there's some reason that you have to do it this coming next week. But for most of you, I believe it would probably be better to wait until after spring break. So I'm getting a lot of emails about, I'm going to be gone. Do I have to take my microscope with me or my bacteria and try to grow them when I'm on vacation? And I thought I've made that clear several times that we're trying to do the microscope labs before we leave for spring break. And we're trying to do grow the bacteria labs after spring break. Worst case scenario, right? You can't start growing the bacteria. You can't get access to the bacterial growing labs until after you complete the microscope lab. So the sooner you get that done, the better. I realize it wasn't up there and available to you. That doesn't mean that you couldn't actually look through the microscope and record your drawings, right, at 10x and 40x, or at least use your microscope or practice using your microscope, just because there wasn't an assignment there. But it's up there now. And like I said, I took the mold portion out because some people have already tried it and they were having trouble growing the mold. So took that out. So it's just 3 slides for you to look at right now and 2 drawings of each of those slides. Again, a microscope quiz that will appear today and will be available from today through Friday at least. So you'll have plenty of time to do that. And then I think what I didn't look at is when our next lecture quiz is, probably for chapters 7, 3, 11, 11 and 12. Is that right? It's 11 and 12. I put 12 and 13 on my paper this morning, but I think it's 11 and 12. All right, so I'm going to minimize you guys for a minute while I check on that. Microbiology. Okay, so now I can share screens with you. I have the right screen up. All righty, so we are up to this bit here, and exam 3 does not have a due date on it. So help me out here. I think that should be, was that supposed to be today? Or did we talk about this last week? Help me out. Kind of.
I don't think we talked about it yet.
We haven't talked about it yet?
I don't think so.
How about we just make this one Friday? Because I don't want to save until after spring break. Can we do that? Let's see. Are there any? Oh, right. I see all of these aren't due until March 29th. So most of you probably haven't even started them yet. Oh, shit.
I haven't seen the Connect up either.
Yeah, it's not it's got March 29th on it from when they, when it brought them over. Let's see when they, let's see if I can see when it.
I don't think they're open for us though to access. I couldn't find it.
All right. Yeah, it's kind of giving me the spinning thing here. Let's try that again. Oh, this might be tragic. All right, so what is this, module 4? Yeah, you're right, they're not even assigned yet. Oh, Dr. Stewart, you are failing us here. All right, so let me just think for a minute, if you don't mind. I probably had thought that we would do this in between. All right, let's see. Well, I'll tell you what we can do. I will open the Connect assignments. And today is what, Tuesday, right? I'll open them for today. They don't let me open them until after the time of day that it actually is. So I won't be able to open them till at least noon, but I'll open them today, the ones that are listed here in module 4. And let's set the quiz for, it'll have to be the Tuesday we get back from spring break. Because I don't want you to have to do it like during break. It's kind of a lot to try to get you to finish it. Like most of you wouldn't want to try to do it between Tuesday and this Friday. So let's open the Connect module for this Tuesday today. And let's say lecture quiz 4 will be, trying to look at a calendar here. February, March. All right, so today's 9th, right? Next week's spring break. So we'll make it, I'm going to make that quiz due the 23rd, March 23rd. March 23rd. And then I'll just adjust everything based on that. So we won't get through quite as many of the disease chapters as we would have, but it's whatever it is, no big deal. So we'll make that lecture quiz 4 on March 23rd. Now, just as a reminder, again, right? So the lab safety, that first Carolina lab module is at the bottom after module 7. And some students haven't been able to find it because it's not in the module that we are currently in, which is 4. And purposely, because I kept all the lab stuff together. I see that the lab safety quiz, I had put a due date on that of the 5th of March. I am going to extend that to make sure that it's still open because there are a lot of people who haven't done it. Oh, it looks like it's still available. I have it available till March 12th. So you can still go in and do it. I'll just move that due date, whatever, I don't care. And then I also wanted to be sure that the lab report that's in that module, I'm pretty sure I put that as April 30th, which is just like the very last day you can possibly turn anything in. Now I have March 19th. I'm going to try to push that. Actually, I'm going to push that closer. I'm going to say March 20th. Let's change that to the 26th. Available until March 26th. So you can still do it. I think you guys realize that, right? So when an assignment has a due date on it. If there's an available until, you can do it until that date, and your instructor will either have, things marked on it if you get point deductions, and then it shows up in our assignments when you turn it in. It shows up that it's the box is plain, which means you turn it in on time, or it shows it late, or it shows it a missing. So you get like a red or like a pinky color or a yellow color, and then you get blue, I think, if it's missing. So you can still turn those in, don't worry. I put no later than March 19th because I'm trying to force you to get it finished by this Friday or next. Right, so that you're all finished with the microscope work before you start trying to grow the bacteria. Because once you grow the bacteria, we are going to be asking you to take another look through the microscope. And if you haven't done the initial microscope lab, that will be difficult, right? So that's why I'm trying to force you. On here, it's going to say it's due the 19th, but like I said, you're going to have until the 26th if you don't get it completed. And again, I am going to add to that module today a microscope quiz, very short, matching, some multiple choice. And that'll be due by this Friday. And then that's pretty much all you have to do this week. Then you just have to get through the microscope stuff, the lab safety and the microscope stuff. And then next week, it's up to you whether you want to work on chapter 7, a little bit of 3, 11, and 12 or not. You can work on them this week. If it was like the regular semester, it would be the same. We'd either have that test right before you left, which would be this Thursday, right? That'd be the last time I would see you before spring break. And it would be rushed because we would just be coming off the last quiz. Or we would have it the day you get back, which would be the Tuesday after spring break. So that's pretty much the same. It's pretty easy stuff. It's mostly just memorizing. So it's a lot of, it's hardly any chemistry. There's very little. There's a little bit in chapter 12 when it talks about the different types of antimicrobial drugs. That probably has the most references back to chapter 9. But other than that, it's pretty much, chapter 7 is very basic. How do bacteria grow? What do they need to grow? What kind of food do they need to grow? It's pretty simple. So I don't think you guys will have a particularly difficult time with the concepts. And again, I will open up those Connect assignments today. And I will keep a due date on them. I will put a due date on them of March 22nd. Right, so the day before the exam on the next lecture quiz, which is on the 23rd. And I will fix that this morning or after we finish our meeting today. And I apologize for that. That's just the semester kind of getting away from me. You'll notice that there's a short answer section to this quiz. It has a time limit of 30 minutes, but it is, it's a scramble of questions. You'll all get different versions of the test, and you only have to you have to answer 3 of the questions. So you'll see 5, and you'll be able to choose which 3 you want to answer, and they're each worth 5 points each. I would encourage you to not just copy the answers out of your book. I've tried to make them so that they are more like a thought question. If you've studied the chapters and you're ready to take the test, you'll be able to answer these questions or be able to find and formulate. And that's 10 minutes a question for you to sit and think about what you want to write. So again, if you've already studied and you're prepared, so I usually give people till the next day on that one. So we'll make it due the 24th. And that's just going to be the standard. It's going to be open for a day. So I should mention that. I'll put that. I did just send an announcement. I'll mention that also in the announcement. I'll send another announcement about these due dates, and I'll mention the short answer part, short answer section. Complete sentences would be nice. Capital letters, periods, whole thoughts. Not like you're texting me, right? But like you're actually trying to write a short paragraph that's relatively well thought out, let's say. So yeah, so I'll fix that stuff up today. I apologize. Like I said, it's just kind of a, like for many of you, I'm also getting a lot of, believe it or not, I'm also getting a lot of, oh, I didn't see that we had any quizzes in Canvas, and I haven't taken any of them. I have my averages an F. What am I supposed to do? So I'm also getting a lot of that this week, because the withdraw date is the 23rd. So right after we come back from spring break. So people are going to have to be making those decisions. As I said, I'll fix up those dates, and we'll move, we'll put the dates for, I'll probably fill in the rest of the semester dates as well during spring break. I'll have a few minutes to sit down and kind of think about it. And like I said, the most important thing to get done this week will be the microscope, the lab safety, and the microscope lab itself, which is basically finding and focusing the 3 slides that are already in your microscope. During spring break myself, I'm going to probably attempt to make a cheek cell slide with the crystal violet, like we talked about, I think in one of the videos. And once I do that and kind of get a procedure for that, I will film it. And then you guys can do that on maybe one of these plastic slides, I don't know, in the kit, or maybe on one of the real glass slides that you received with your microbiology, your real microbiology stuff. So let's think about that for the future. It's hard to get these back in here. There.
I just have a question.
Sure, Hannah, what's up?
Can you just show me where the microscope labs are?
Yes, so they're at the bottom. All the lab kit labs are at the bottom, and you have to read through you have to read through the safety manual, take the lab safety quiz, and then this document is, this is the whole document of, you'll have to download it and then open it yourself. I already have it existing somewhere on my computer, but I don't know where right now. So yeah, just open it. I think I can open it in Word. That's just the booklet that came with your microscope.
Mm-hmm.
If it's not, this looks like it's not working. If it's not there, go to Carolina website and just type in fundamentals of microscopy, and it's just going to give you there's a little booklet. Did there not come a booklet with in your kit for the microscope itself?
No, I did.
Yeah, I think it did. I had a little booklet myself that came with my kit that actually had like pictures of the microscope. It wasn't like the other ones. I don't know where it is right now. You didn't get a little booklet that opened up?
No, I did.
No?
No, I did.
You did. Okay, so
yeah.
And it talked about making a little brine shrimp. You got a little package of brine shrimp to hydrate and look under the microscope with little slides, right? That's what that document is, that PDF that's not opening. That's what it is. It's that booklet.
Okay.
So,
I took the lab safety quiz already.
Right. So you have to take the lab safety quiz. You have to actually do the lab report, which I just opened this morning. And there's also going to be a short microscope quiz.
Yep.
That will be put in there today, and you'll have until Friday to complete that. And then after you've finished all of those, then you'll be able to open up the second lab module, which is right underneath it, which is all this, the giant one here. Right. So this one, and you don't really want to open this one until you're getting close to ready to start growing your bacteria. So you can certainly open up the planning document and look and see like the dates and how long you think things might take. And like I said, if you want to start working on it over spring break, that's fine, you can. I'm just going to change the lab report date from December to April 30th. So these are all the labs. I have them all as one document right now because it was just easier for me to grade that way. And I always think I'm going to change it into multiple smaller documents, but I never seem to find the time or motivation. So I'm just going to change these to being due April 30th, just because I don't want to grade them all. So they're actually already available, but we'll just put from today. Right. So I just don't want to grade them all like March 2nd because people turn them in late, you know, which yes, they do. So there, now you should be able to access that. And again, like I said, I'm going to pop a little microscope quiz in there. And then after you complete the microscope quiz, you'll be able to, and that lab report, you'll be able to open up the the second one.
So but I'm not gonna start the Carolina kit till after spring break, right?
Right, but you're going to want to open this, and I would open this and watch the rehydrating videos, and I would look at the planning document before then, because it's kind of it's rather complex, right? And it's very multi-step. So you have to get yourself some bleach, some ethanol, a Sharpie, a lighter, some matches, right? You have to collect up all this stuff as well as then start growing the bacteria. And then it's going to take, like this says day 7. It'll take longer than that. It took us about 12 to 14 days. So this is like ideal if everything works perfectly. And this day 4 here, when you do the simple stain and the gram stain, that's a long day. That's like over an hour just focusing on making the stains. And then whenever you have time to look at the stains with your microscope. So some students wanted to separate that out and spread that out over 2 days because they didn't have the time to do it. So this is just a sample schedule that I made up, but you can certainly modify it. And you may want to start watching those videos, kind of help you decide how much you personally want to do in a day. I will probably be adding, not in the module we're doing right now, but probably in module 5, I will add a couple more virtual labs to help you a little bit with the preparation, because the early labs will be the early growth culture labs that you guys have to do. First, you have to rehydrate your cultures, and then that may take a day or 2 until they're grown up enough. And then you have to transfer them and do isolation before you can do the staining procedures. So the staining labs you've already done virtually, but you haven't done the isolation and the, I don't think I did. I know I didn't put those in yet. The isolation technique and the transfer technique labs. So I'm probably going to pop those into either the module we're in or the next module, or I might put them in twice so people see them. And it would be good if you could give those a whirl before you actually do the real lab. But you know,
So once you add those, I already did all like the virtual labs, so I should go back and then do the ones you added, right?
Yes, and there's going to be connect assignments for this next module. There's quite a few here. I broke them up into little tiny bites because chapter 12 is long and boring, and if you have to sit and do the whole thing at once, you'll go crazy. Okay. And then you'll notice after that, after chapter 12, there are not very many connect assignments. I think I put some in for some of the disease chapters, but then the connect pretty much ends except for some more of those virtual labs that I'm going to, like I said, I'll probably add those to, because we're getting close to actually doing them. It's tough with spring break, like right in the middle like this, like right when we're starting to, like, I want to do all these things that, you know, we would normally just be doing, but anyway. All right, virtual labs. I'll look for an isolation lab and a transfer lab. Minimum. So look for 2 virtual labs at the least to be added. And I will probably put them, again, I'll probably put them in twice. I'll probably put them in module 4 and module 5 so that people see them, right?
Okay.
All right. Okay, other questions about the material? Other issues you're having, or you're good?
No, I just want to recap.
Yeah.
So recap. Micro quiz Friday.
Yes, between today and Friday. Yes.
Okay, so
And yeah, it's going to be easy. I mean, and
okay.
I don't even know if it's going to be timed. So I need you to know the parts of the microscope, because now it's pending doom that you're going to actually have to go back to class and use a microscope.
Yeah, I thought
And
for some reason we already had a microscope quiz, but then we didn't. So I was like, oh, okay. So
it was originally, I had it buried in that microscope lab.
yeah,
I just took it out.
That's what it was. Yeah.
Yeah, I just took it out. I'm going to just make it a standalone quiz, because what happened last semester is students didn't click on it. I don't know why.
What?
They were like, oh, I didn't see it. I'm like, well, it's in blue, and I thought you knew to click blue things, but whatever. So I'm just going to make it a standalone assignment, quiz, whatever.
And those virtual labs, when will they be due? The 2 that you're adding?
I will probably put them, I didn't write a due date on those. I'll probably put them like March 24th.
Right before the quiz. Okay.
Well, right after the quiz.
Yeah, you're right.
Right. So I'm going to put that on here, March 24th for the virtual labs. Virtual labs, okay. Circle that in red. So if I open the Connects today for this module and have them due on the day before the quiz,
22nd.
The 22nd, and then the lecture quiz on the 23rd, and then I'll make the virtual labs due on the 24th. But okay. I'll open them today so you can do them whenever you want.
Mm-hmm. Okay, so that's all my questions.
Yeah, and then there might be a couple more virtual labs. I'm not sure. I haven't had a chance to look at them yet. That sort of supplement the biochemistry labs at the end of the second module, the biochemical testing. So I might pop a couple more in here that would be due like April 30th. But those I'll put like right before you do that exact lab. So, but I would like you to at least practice transfer and isolation before you actually start growing your bacteria. So, like I said, the isolation and transfer virtual labs will appear in many modules. They will probably appear in module 4, module 5, and then I'll probably also put them at the sort of close to the top of the lab kit module 2. So let me write that down too, because I already put module 4, module 5, and lab kit 2. Lab kit 2. There. All right, my little paper is getting kind of messy here. I better rewrite this. All right. Yeah, anyway, I can do that later. All right, other than that, questions about content for chapter 7, the little bit of chapter 3 that we go back to, chapters 11 or 12.
Just a quick question for the labs, for the module 2 with all the like planning and the step-by-step, that's what you're modeling and then you're gonna post is those steps basically, is that right?
Right, so the sample plan was just to kind of give you guys a schedule, like best case scenario schedule of how long it would take you and basically what activities you would have to be doing every day, right? And then this, of course, I planned this out just like you would before I actually sat down and tried to do it. So when you actually go into that, it seems confusing at first, but it's not that confusing. I get it. It's like a whole new thing at first, right? So all of the directions for the Carolina labs for exercises 2, exercises 3 and 4, 5 and 6. So the parts, the labs you actually need to do with the bacteria. These are all of the directly from Carolina. These are the directions, like the full directions, right? It's like, this are the supplies you need. This is how much time it's going to take you, right? As if it was like a lab book, right? It tells you exactly what to do. Let me go back.
Gotcha. So those are the steps where we just like go and look for there. Like when it's time for like a biochemical test, we just check the pamphlet, the whatever out and go from there.
Yes, and I made videos for each day. So
Okay.
the ones that are labeled video, and like this one that's labeled video Carolina Lab Kit Rehydrate Cultures, I think that's me doing the video. Yep. Right, so I would take a look at those. So I've got them for every day of that module, of the lab module, right? So this is Lab Kit Video Day 2, aseptic technique, which is exercise 2. So I would read the experiment, I would watch me do it, and then I would do it myself. Like, then you do it, right? And then for Lab Kit, for exercise 3, again, I tried to keep them in the same order. This is the Carolina instructions, like your lab manual or your lab book. This is me doing that exact lab, right? And then this is the next day, me doing the bacterial smears, exercises 4 and part 1 of 5. And then this is me that's, you know, those days, the ones that are labeled by day, except it looks like I didn't label the first one by day, right? This is just me rehydrating the cultures to get you ready. And I think in this video, I think I reiterated how to make, there's an alcohol solution sitting there and a bleach solution and how to light your little burner, right? I'm not sure how far along I am in this particular, where it happened to freeze. But all of those sort of like setup things you'll find in, most of them are in this Rehydrate the Cultures video.
Okay, thanks.
Yeah, it'll take a little, like I said, it'll just take a little organization for you individually, and it's going to be different for each person. You just have to kind of watch and get all the stuff out in front of you and then be like, okay, what do I need to do, and get yourself organized, right? So it's a little planning involved. Yeah, and I think this is a lie. It wasn't really day 4. It was like day 9 or 10, right? So it was, but I just tried to keep it as if it was just happening one day after another. Right? I don't think I recorded. I think I recorded another one at the very end last day. Yeah. But between Lab Kit 4 day and the last day here, that was either 3 or 4 days because my cultures were growing very slowly. But for you, now that it's warm again, you know, or going to be getting warm by the end of March, you know, it may only take 24 hours, 48 at the most, like a day or 2. Ideally, you would be able to complete all of this Lab Kit Module 2 in about, I would say, on average, between 6 and 10 days. 6 being if you're just really diligent and everything's growing great. 10 days if something you have to wait, or you missed a day because, you know, you were like, oh, I can't. But you have to keep the bacteria growing the whole time because you need them for all of the labs. Once you start growing them, once you start rehydrating the cultures, then you have to keep them alive for all of these other techniques. So that's the important thing. Don't throw anything away until the end. All right, so that thing is not working, whatever it is. That fundamentals of microscopy. I'm just going to get rid of it. There, it's gone. I don't know where I got that. I'll have to see. I'll have to try to trace that back where I got that from. And then I'm going to pop a quiz in there for the microscope. All right, anything about the content from the module 4? All right, so I said this was going to be March 23rd. Done. Tuesday, there we go. There, now everyone can see it sitting there at the bottom of the module. All right, and I will work on changing the Connect assignments so that they will open today. Connect for module 4. And then they will be due on the 22nd of March. I just fixed the lecture quiz 4 is going to be due on the 23rd of March. So that's Tuesday. And then there are a few virtual labs that I'm going to add, and those will be due the 24th. Isolation. I'd like you to see and practice isolation and transfer before you actually have to do it. It would be good for you. So I'm going to try to find those two and pop them there. All right. Anything else? Are the bits of chapter 3 that are going alongside going to be reviewed? No, it's the parts that we did not do from chapter 3. So I think if you take a look at the note outline for chapter 7, this is the lecture for 7. So I would normally in class, oftentimes I just pass this out or I tell students ahead of time, I put up a note outline so you can get it and type it in, right? This is just going to help you because again, chapter 7 is I find it kind of long and boring. So I try to do something to make it a little more like you're a little more engaged. So as this starts with chapter 7 and it goes through all the different types of molecules that cells, bacteria need for nutrition, and then the different types of organisms. There are different types of nutritional categories, right? And then it goes through like, what do they need oxygen for? Where do they get oxygen? What do they need phosphorus for? Where does it come from? And then this is the part of chapter 3. So it's pages 56 to 61, and it's mostly about the media. And the reason we bring it back in now is because for bacteria, when we're growing them in culture, of course, the only way they're going to get food in the laboratory is if we provide it for them. So I find that this part of chapter 3, which talks about media and how much water to use and whether it's hypotonic or hypertonic or isotonic, like, oh, that really fits well with chapter 7 because we were just talking about bacteria need carbon, they need salt, they need sodium, they need chlorine, they need oxygen, and it is provided in the media that we make, right? Or in the media that was shipped to you in your kit. So this portion, the first part goes through the different types of media, which actually you guys have most of them in front of you now. And then how we use media in the lab. And again, and then that's it basically. And then this part is how you can use the media that you guys received, both the nutrient agar and the trypticase soy agar, the TSA agar in the Petri dishes. Those are both general types of media or general purpose media. And both of them actually are what we would call complex, right? Actually, I think because they have like peptone in them or tryptone in them, which means, so trypticase soy agar means soy proteins have been digested with trypsin. And we don't know exactly how many grams or milligrams of each of the 20 amino acids are in there, but we know that protein has been digested and it's in there, right? So that's why it's considered complex or complex. That's this part, right? And then these types of media, the selective and differential, in the test tubes, the triple sugar iron media is the only media that we could provide for you in kits that got shipped home. But normally in the laboratory, we would use a lot of selective media. We would use some different types of media to grow different types of bacteria to help us differentiate between all the different bacteria just by using based on what they eat and what byproducts they make. And then this part should be review, diffusion, active transport, passive transport, and then some mostly vocabulary, right? Having to do with oxygen requirements, as well as temperature requirements, as well as pH requirements, pressure requirements, and then just some very specifics about how bacteria multiply, how we count them. That's the end, methods for counting. And I didn't put any part of the outline. The outline's kind of ends there because you can just watch the video and add notes. So yeah, I would watch the videos. That's always my answer, right? I would watch the videos in module 4 very carefully with that note outline, and then just take the notes. So it looks like 7. I've got 2 videos for 7. That includes the little bit of chapter 3 in the first one. And then there's a video for chapter 11 that looks like it's an all-in-one. I just muscled through it. That's a long one. And then there's also a video for 12. Actually, maybe not. Where's the video for 12? There it is. Antimicrobial therapy. So yeah, and then some homeworks. This one I might move into the next module as well. I think I put it in there twice. No, all right, whatever. All right, so then, yeah, good luck and enjoy your week off or not off or spacing out your assignments, whatever you want to do. You want to wait until, the end of next week and then just try to catch up on all of this chapter 7, 3, 11, and 12, be my guest. I don't feel too badly about putting it off because I felt like it was a lot for you guys to get through up through chapter 9, and you did kind of have a little breather this week. So I probably should have pushed a little further, and you wouldn't have as much to do over spring break, but we'll be fine. We'll get it all done. Don't worry. All right, other questions, comments, worries, concerns? Yes, no, maybe so. All right, stop sharing. Stop.